I debated what topic I should do for my second talk, and I decided on this. It's a bit of an experiment. So I work on this other system or question in my lab where we're actually interested in a practical problem, and that's Lyme disease, and because it's a big problem in the U.S. And I think I guess because you reach a certain age, and I talked to Christian a little about this because he's a similar age to me. He's probably much younger, and you kind of like maybe think, oh, geez, I feel like I should work on an applied problem for a bit before I die. But it's an interesting problem because something happened by accident, which led me to believe that some of the tools we've developed for synthetic populations and some of the ways we think could actually be used to deal with this problem. So although we're certainly not evolving these mice or anything, I think I'm going to try in the talk to sort of draw parallels to the work we're doing in Drosophila and yeast in experimental evolution, and how some of our ways of thinking and modeling could be used to address this problem. So like all the talks, feel free to stop me anytime, or we'll probably finish early, or don't ask any questions if you want to have lunch early. So again, the title of the talk is Understanding Lyme Disease Host Dynamics Using a Peromyscus leucopus Synthetic Population. So Lyme disease, I don't know if everyone knows what it is in Europe, but it's a disease in the US that's caused by a bacterium, and the bacterium's called Borrelia burgdorferi, after the Burgdorfi, who's the discoverer of it. And it's, for those of you that know it, it's actually transmitted by a tick bite, right? So you go out on a hike or something, and you're bit by a tick that's carrying this bacteria. This bacteria does not want to be in a human, really. We're a dead end. But they can live in us fine, and they cause a number of health problems. Initially, if the tick has Borrelia and it successfully infects you, you get fever, headache, fatigue, and you get this characteristic rash that forms around the bite that's like a little red halo that grows out. When we see this, we can treat it fairly aggressive with antibiotics, and many people respond. Some people don't. Also, if left untreated, there's problems, and there's debate about how severe these problems are, although I think in the US we're generally becoming more concerned about it, and they seem quite serious. And these include heart problems, nervous system problems, and especially joint problems where you have arthritis-like symptoms in the joints. Okay. Right now, there's about 3,000 cases per year in the U.S. But what's freaking people out is that those cases are increasing in frequency. Some of that increase might be due to more awareness of the disease, so we have better reporting. But definitely, some of it's due to changes in incidence due to global warming. We like to say everything's due to global warming, although maybe even a bigger problem in the U.S. might be due to the way urbanization patterns are happening, where people are moving into these semi-rural areas where there's especially deer and Peromyscus, and so they're exposed to the tick more often, although certainly both things are contributing. These are the ticks. This is the younger life stage. This would be an adult that you can actually see if you're bitten by a tick. Many of you have seen on your dog. There will be pictures of ticks in the talk, and I'll try to tell you to avert your eyes for some of them, okay? Because they're kind of creepy if you've ever seen them live. How many have seen them on their dog or something? How many have had a tick bite on themselves? Oh, wow, okay. Where? In here or in the U.S.? Yeah, so you don't want to get a tick bite, especially in this area of the U.S. So where it's especially prevalent is in the Northeast. The term Lyme comes from the little town of Lyme in Connecticut, I believe, which is like a rail stop north of New York City. So rich people live there and commute into New York City on the rail. And it's especially, if you look here, it's like incredibly dark blue in this area here, which is Nantucket and Martha's Vineyard, which is some of the wealthiest real estate in the U.S. So it's like where the Kennedys have like summer homes and like bankers and that give like $1 billion to Harvard to have a statue of themselves until they take it down because it turns out they were a racist 50 years later. Okay, so but that's where so there's a lot of, not only is there a lot of interest in Lyme in the U.S., but there's a lot of rich interest among wealthy white people. The other place, kind of, you can see it here, is around San Francisco, right, where we've actually gotten money from something called the Bay Area Lyme Disease Foundation. So there's a lot of rich Silicon Valley entrepreneurs very interested in Lyme disease. So it's kind of neat, right? Because I think not only is it important to see if we can take some of our ways of thinking and apply it to problems, but it also can be a source of funding. You know, we're academics, so anyway, we always compromise our principles eventually. So a lot of people don't really understand how it works, but so the ticks are called deer ticks, right, that you get. I mean, that's the other name, the common name they go for that I grew up with. And what's interesting is that the Lyme is not really in the deer, and the deer don't transmit the Lyme really at all, okay? What the deer are is a reservoir for ticks, right? So deers support a lot of ticks and lead to large tick population sizes, okay? What normally happens is that the younger stages of the tick tend to bite, we think Peromyscus, especially Peromyscus leucopus, is one of the better hosts, but other small critters too, which I'll show you some data on, these animals look like they have had a long-term coexistence with the ticks and the bacteria, such that the bacteria are actually specialized to reproducing in them. The ticks bite the Peromyscus, and in a perfect world, they grow up to a nymph and they re-bite Peromyscus, and that's their life cycle normally. But occasionally, the tick by accident bites the human, which is a dead end, but that's the problem, the public health problem that we're worried about, right? Okay, so that's why it's a thing, right? Okay, but again, Peromyscus seems to be the main host. How do we know it's the main host? Well, it's neat about applied problems. There's often lots of data, right? So this is a chart where we have where different kind of, I don't even know what the term for these are, but small mammals that are potentially infected. There's been studies of this. So we can look at the proportion of infected ticks that bite one of these hosts. So it looks like if a tick bites a white-footed mouse, which is the other name for Peromyscus, almost 100% of the time they're infected with the Borrelia, and that's less, so it's transmitted more poorly from these animals. And if you actually look at the actual titer of the bacteria in an infected tick as a function of what they bite, the actual titers that are transmitted is much higher when they're transmitted from this species than these other ones. Now, it doesn't mean there aren't other Peromyscus species or other hosts that are good, but it really looks like the favored host is Peromyscus, and especially Peromyscus leucopus, we think is the dominant species, right? So that's a species we want to focus on. What are some strategies for dealing with it, right? Well, we could reduce, one of the old strategies is reducing deer populations to reduce tick numbers, right? This is a wholesome American 12-year-old girl who's killed her first deer. So that's one way we could do it. People moving to urban areas don't like this idea. I had some excellent venison sausage last night that the people that didn't come to the Harkinger missed out on, but there is some advantages to this strategy. And it can reduce tick numbers, and it is being used. But I think it's something we should be aware of when we move to these urban areas. Sometimes hard decisions have to be made to keep those tick numbers down, right? So another thing we do is vaccinate humans. So actually there was a vaccination for Lyme disease that made it all the way to phase 2 trials in the US, and it showed some efficacy, and then the drug companies just decided not to push it. And the main reason is that in the US especially, there's a huge resistance to vaccinations, where kids have those that have kids know they're vaccinated to about 20 or 30 things now. How many shots have your kid? Your kid's still not even year 3 yet, but up to 25 or something things, it seems like. Yeah, and obviously there's a group, especially again, of wealthy, educated white parents who don't want to vaccinate their kids, and so I think, or get vaccinated in general. So the drug companies abandoned this as something they could make money from. But then they did actually carry the product through, and you can actually buy the exact same thing that was being tried to vaccinate humans to vaccinate your dog or your cat. So it's kind of a neat case where the product that was supposed to be used in humans is actually being used in pets, because we don't have nearly the same qualms about vaccinating our pets, apparently, as we do our children. And we could give a whole talk about this. So another idea, so one thing that's been highly studied that I'm not even going to talk about is to attempt to alter the ticks, right? So that's an interesting target. We could go in and try to do something to the ticks to make them poorer hosts, right? I'm not going to talk about that, but people are pursuing it. It's a pretty good strategy. And the final thing is to try to do something about Peromyscus, right? So we could kill Peromyscus would be one idea. We could try to make Peromyscus a poor host, or something we actually have granted to do is we're going to try to vaccinate Peromyscus in the wild with basically these like food drops of this vaccination, and then try to make the Peromyscus resistant to the Borrelia. But it's hard to get, when you're putting food in a food drop, it's hard to get efficacy up, right? The big thing that's being discussed in the US, and there's been a number of articles in the New York Times and the popular press, and I even think I have a YouTube video link in here some way, is to try to do gene drive in Peromyscus. So this is as far as so gene drive's being discussed as a way to do malaria and other mosquito-borne diseases. As far as I know, this is the only case where we're talking about a mammal, and there's actually discussions pretty far along to basically introduce this CRISPR system into Peromyscus, introduce them into the wild, and that could be used to treat the disease. So the idea of a gene drive is that we introduce a construct that has the Cas9 gene and a sgRNA that basically can cut the target, that can cut the Cas9 out, and then that basically that construct will make copies of itself and result in non-50-50 offspring ratios. And there's some mathematics that would suggest that if you introduce these gene drive things into a population, they'll increase in frequency, sort of like a PCR reaction, so that a large fraction of the animals that will eventually be infected with this construct. And so then if you also put in here something like a monoclonal antibody against the Borrelia, that could result in resistance, right? Although, again, there's a lot of work being done on these strategies, and it's looking like perhaps the gene drives aren't as efficient as people think, because you can get these non-partial repair events and other things. And in fact, there's people, I talked to someone yesterday working on this here, right, Anna, right? Yeah, right we go. So it's not, this is a fairly, how many people know about gene drive? It's a lot. So it's pretty, okay, so it's I mean, we're teaching it now to undergrads in genetics, I think, so it's sort of become a big idea pretty quick. Okay, so, and in fact, this being discussed, so the idea is to build heritably immune mice. Could take a couple of years to engineer these. It's being discussed initially to release these on islands, but I don't know what the gene flow is on and off of islands, so if this thing gets released in the wild, it could quickly, spread throughout all of North America. What's interesting is there's even public forums going on in that you can follow that link to see. So it's a fairly surprisingly being releasing these mammals into the wild is being pretty highly discussed. I would argue that we probably, before we do that, we should know a little bit more about the biology of Peromyscus, and so that's one of the reasons I got into this. This is just a picture of Peromyscus. So unlike all the other talks, these things have brown eyes that look back at you, right? So there's a lot of, that creates a lot more rules for working with them and some stuff, but this is the system. And what's interesting about Peromyscus is that although it's called a mouse, it's hardly a mouse, right? So here's it's my only phylogenetic tree. This is a simple tree I found that I unfortunately didn't cite, showing the phylogenetic tree of some common mice-type things we know about. Here's mus, or the house mouse, lab mouse, and rat. So you can see there, actually, despite us calling Peromyscus up here a mouse, it's actually not that related to a house mouse at all. In fact, a rat is much more closely related to a house mouse, although you wouldn't know that by looking at them. And Peromyscus is more closely related to things like hamsters and gerbils, which you can see over here, but they're not even that closely related to them. So there's all these like mouse-like rodents that are quite diverged from one another. So we think that Peromyscus and the rat/musk lineage maybe diverged 25 million years ago. And one thing that's come up in these talks here is that like the mus and rat are probably diverged only 10 to 15 million years ago. So like it's kind of weird when population geneticists throw around 15 million years, 10 million years for equilibrium dynamics to occur. To me, it always just freaked me out that we're talking about population of genetic events that take as long as species are old, right? So it's kind of weird when we look at this. But the point is that we can't, so our feeling was we couldn't really these are diverged enough that although mus is being used as a study, and certainly lots of mus have been injected with Borrelia, if we really want to understand how Borrelia works, really, and their biology works, we probably have to do experiments in Peromyscus because they're quite diverged genetically and genomically. We can't even really use the mouse genome because these are pretty diverged species. So it's kind of an example where I got into this, and although I'm a population geneticist, we sort of decided the first thing we had to do was actually do a genome assembly for this species, and I'll talk a little bit more about that. And so we did, and it was published this year, I think, and it's the first Peromyscus genus assembly, so it was somewhat important for other reasons. So this was an assembly using a combination of PacBio. Could be done a lot easier and cheaper now, but we had about 36x of PacBio data, 54x of Illumina data, and then 2 Hi-C libraries. So these are these libraries you can use for scaffolding. Based on the the PacBio and Illumina data alone, we had an N50 contig length of about a little less than 4 megabases, so that's actually quite good. And then we could use basically the Hi-C data to scaffold these contigs, which results in a final assembly of, let's say, 2.5 gigabases for giggles, so again, this is like the same size as a human genome or a mouse genome, of which it looks like about 97% of the genome and something like 98% of genes are in these chromosome-linked scaffolds. So this is a pretty good assembly by modern standards, I think. Okay. We can look at Synteny with mouse. This is commonly done in mammals. This isn't that interesting except for a few technical points here. One thing we observed is relative to the linkage maps in this species where they had a chromosome 8. Our chromosome 8 falls out as 2 different chromosomes, which I think Hopi Hoekstra has also seen in her unpublished assemblies of Peromyscus maniculatus and Polymorphism, I think is the other species she's sequencing. And chromosome 16, what the previous linkage studies had called chromosomes 16 and 21 are actually a single fused chromosome. A little warning about scaffolding with Hi-C. You could read the paper, but it can tend to assemble it accidentally creates inversions in a lot of your contigs. The other thing it tends to do, which you can see a little here, so when you look at the Synteny map, if you look here, like you see a little bit of blue at the end of chromosome 2, so that would be chromosome 6. You see a little bit of, my things moved around. Oh yeah, you see a little bit of this red here at the end of chromosome 1. You see a little bit of orange here at the end of chromosome 5. You see a little bit of red here at the end of chromosome 7, a little bit of, is that brown here at the end of chromosome 19. So this Synteny map makes it look like we're getting these telomeric translocations. And in fact, we provide some evidence that this is a common artifact of high C assemblies where it tends to shuffle telomeres. So you got to be a little careful if you're doing high C scaffolding. That's going to apply to probably most people in the room, but it's something that the field, I think, is going to work on. That it's so these scaffolded assemblies are a pretty good starting point, but they're not. I wouldn't say they're the same quality as, say, the human assembly, right, or the mouse assembly. It's probably as good as rat, which isn't a great assembly despite being a major organism. We did a lot of work to take the Peromyscus assembly, annotate it with genes, align it to mus and rat and humans, and then it's all available as in Santa Cruz browser, so you can go in and you can look at our assemblies. It's all downloadable and all that. Of course, this is just an example where I typed agouti into the search window, and I've set all that up, and here's what agouti looks like in its genomic context. You can see how it aligns to mouse. You can see some SNP and variants we've annotated. Of course, like a mammalian genome, there's a lot of transposable elements and repeat elements in it. So the point is, if you're interested in some gene, you can go into the browser and download that region, design primers, look at splice variants, all that stuff, right? So it's pretty useful. Agouti is kind of one of the genes that's pretty well studied in this system that many of you probably are familiar with Hopi Hoekstra's career, looking at, or part of her career anyway, looking at coat color evolution in the genus, where it's a famous story that Henrique thought I should have talked about the other day, where you have these, they normally have that brown coat I showed you, but these Peromyscus that live in sand dunes have white coats, because then the birds don't kind of see them and catch them, like raptors. And that's due to mutations in this. She showed it's due to mutations in this gene. And in fact, a lot of coat color mutants pop up in this gene. So the better genome allows us to do stuff, right? So which we probably couldn't do that easily before. We've found doing RNA-seq with just a transcriptome is problematic, and we much prefer a genome. And so I kind of feel like the talks in this meeting have really, there's been a lot of talks about actually, obviously bacteria, which are have complete genomes annotated really well. Yeast, arguably the best eukaryotic genome in terms of knowledge and annotation. Drosophila, extremely well annotated genome. C. elegans, also one of the big models, right? So a lot of the work we're doing in Evolve and Resequence relies on working in these systems that other people put a lot of effort into. And so if you're going to go off the reservation and work on some species that's biologically really interesting, one of the first things you have to do is get your system up to that model system standard or semi-standard, right? So we did some experiments where we can infect the naive Peromyscus with Borrelia. How you do that is you just take a mouse that's infected, take a little bit of blood, and then you infect the naive animal with that blood from a donor, right? So it's a pretty easy experiment. After about 5 weeks, the bacteria or spirochetes are cleared from the blood, so you don't see them anymore in the blood. But you can actually, if you do an ear skin punch, you can see the bacteria, living bacteria present there. And then we can do RNA-seq and look at what happens. And I'll show you that data. Oh, wait, this was one of the, so if you're grossed out easily, don't look. So you can maybe see why the spirochetes end up going to the ear and being alive there. It's a very easy tissue that's not protected by fur that the ticks actually prefer to go to and eat, and that's what they do. They target that area. So if you're the bacteria and you want to do well in your host, what you want to do is get into the bloodstream, and you don't get transmitted from the bloodstream. You got to get out to where the ticks are going to bite to get through your life cycle, right? So the fact that we see the bacteria like going preferentially to these areas of the animal where where they're going to complete their life cycle is evidence that they have a long-term relationship with the species, right? So in us, if we get bit, as far as I know, the Borrelia don't go to our ears, right? They go to our joints, which is undesirable. Probably be okay if they went to our ears. Okay, so, and consistent with that, we see, if we just look at RNA-seq data, and I'm not going to tell you stories about genes or anything, we see very few genes that show changes in gene expression in the blood 4 weeks after infection. The blood looks relatively normal, whereas the skin is still actively involved in some kind of complex dance with the bacteria, okay? So there's still a lot of changes in gene expression in the skin 4 weeks after infection, yeah.
Since the ticks technically suck blood, is their presence in the skin then more of a host evasion thing?
It could be, yeah, I don't know. Well, so, although I'm not going to talk about it, it looks like the reason, so if you go out in the Northeast and collect Peromyscus in the fall in the US, almost every animal you collect is infected, and they don't seem sick. So what's interesting is that unlike us, the Peromyscus don't get sick, and it looks like one of the ways they're doing that is they're just kind of ignoring them. They're not mounting an immune response against them. So our problem isn't the Borrelia in our joints, it's our immune system trying to attack them, and they're able to fight back. So then we get inflammation. So yeah, that's what I know. So my collaborator is actually one of the co-discoverers of Borrelia, so he's a lot better at the biology than I am. I'm learning it on the fly. You'll see I'm the genome guy or the population genetics guy, but we'll get there, okay? So so the point is that there's definitely stuff going on where there's a host interaction with the bacteria. So again, when I do the yeast experimental evolution, it's like really simple system, right? And then this is really fun for me because it's like, geez, there's like a host, and then there's a bacteria, and there's an interaction, and there's transmission. And so it's like when you go into these applied problems, there's all sorts of really interesting and fun biology, right, that sometimes we don't get in our simple systems, or we ignore. I don't know. We could talk about that, right? So it's kind of a fun, it's a way more interesting system than it gets credit for. So let's go back to the Peromyscus genome sequence. One thing we observed that I don't think people realized is that Peromyscus is highly polymorphic at the DNA level. So this maybe is not much of a surprise. My collaborator, Alan Barber, claims that it's the most common mammal in most places in North America. I suspect he's wrong, and it's a bat. But anyway, the point is there's a lot of Peromyscus, so their effective population size is probably quite large. In the single lab individual that the reference genome is based on, we could align our 72X Illumina reads back to that genome, and we identified about 10 million SNPs. So this is a lab individual that's somewhat inbred, a single individual that's somewhat inbred that we align reads back to, and we identify about twice as many SNPs that's known in all of the history of human research, right? In 26 individuals that we low-pass sequenced, we identified 42 million SNPs, so that's like an order of magnitude more SNPs than's known in humans. And these are only SNPs we saw in more than 2 individuals, again, sequenced to 2 or 3x. Here is the evidence, at least in the sequenced individuals, the SNPs are real. When we align the reads, if we look at the frequency of the SNPs in that individual, they tend to peak around 50% with some variance, which is what a SNP should look like in a diploid individual, right? Because if you just look at coverage, right, a heterozygote is like half one allele and half the other in short read world, right? So in fact, based on this number of SNPs, the 42 million SNPs, which is undoubtedly an underestimate, it suggests that about 25% of randomly chosen 20 base pair CRISPR guides, so if you were going to design a gene drive thing that used a CRISPR guide, are likely to harbor a common polymorphism, and that common polymorphism would basically be a resistance allele to the CRISPR drive. So it's extremely likely that in this system, if you were just choosing CRISPR guides at random, you'd have resistant alleles at intermediate frequencies in the population. So that's already a value of this resource. In the wild, LD drops extremely rapidly in this species, although it's not measured that well. But by 25 kb, it's approaching background. So in wild-caught animals, at least in the population we've studied, something like GWAS is not going to work because you'd need SNP chips with on the order of 40 million SNPs, and then you're still probably not going to saturate in terms of linkage disequilibria. So that's an important, again, an important practical point. Okay, so that's what we know about the genome of Peromyscus. So now, like my last talk, I'll have a brief interlude here. I showed you I'm not that into art, but I showed you a painting from 1950 that's here in Vienna from Jackson Pollock, and actually some students commented on it. So I thought I'd show you a picture, one of the older paintings here, or one of the older famous paintings here in Vienna. This is in another museum, and it's the painting in the Peter Bruegel, what they call the Peter Bruegel room, which is a room full of Peter Bruegel paintings. And that's a painting from 1565, so it's quite an old painting. And we were wondering what the most valuable painting is in that museum. It could be this one, but it's probably the Vermeer, right? Because there were several copies of each of these made. But anyway, I thought Christian would help me out on this. But I actually put this in to make me stop and talk for a second about something. So so the whole first part of the talk here was just about assembling a genome, and it's like that has nothing to do with this course, right? Because we're interested in figuring stuff out about complex traits or experimental evolution. But I do think that's important that sometimes, like if you want to work in a non-model, this is a step these days. We just have to find the resources and suck it up and do it. It's a lot easier than it was 10 years ago, especially with PacBio and Hi-C data, and it's just a thing that sometimes you have to do. And so I kind of went off track and did this, but I think it sets us up to actually do some nice work, okay? Now, so a little bit more history. The other work I could have talked about today is something I'm pretty well known for, called the Drosophila synthetic population resource. It's a thing much like the synthetic populations of yeast we talked about the other day, that we have. We started with 8 founders. So these are highly characterized isogenic lines. In fact, the founders of this resource are now completely de novo assembled, and we know about all the structural variants and TEs in them as well as SNPs. We basically mixed these founders, allowed them to recombine for several generations, and then made a set of actually 2,000 inbred lines. So investigators order these lines up. They measure them for some quantitative trait, and they can basically map QTL to sub-centimorgan intervals. So if we look at this resource, this is sort of I think this is the X chromosome. So this is genome position here on the X. This is the different recombinant inbred lines here. These are the colors are founders, so we can see, like you'd predict, the chromosomes in the Drosophila synthetic population resource are basically these recombinant haplotypes, where they're recombinant between these different sort of flavors of founders, and that's the thing that allows us to map quantitative traits. And so my experience with this synthetic population resource is kind of what led me to basically try to do this, or to collaborate with Molly and basically make these same things in yeast, right? These same synthetic populations. This is just an example, it's a fun example of a QTL we mapped. We mapped many QTLs for many traits. So have other people using the resource. This is a QTL. So what's neat is we can actually give Drosophila chemotherapy drugs. Especially if we give females chemotherapy drugs, we can give them these drugs at much higher doses than humans tolerate, like in terms of megs per pound or whatever. But what these drugs do in females is they knock down oogenesis, because oogenesis in flies basically involves rapid cell division, which is exactly what chemotherapy drugs target. So these are 2 chemotherapy drugs, gemcitabine, which is used, I think it's still used as a frontliner in breast cancer. MTX used to be used in childhood leukemia, not anymore, although it's still widely used, especially in Europe, to treat arthritis. So if someone has arthritis in Europe, they are often taking MTX. In the US, we use more expensive drugs. It's not clear they have a higher efficacy, but they're the so-called bio drugs. Anyway, so we map a strong QTL here where we have a candidate gene and everything that we think could be contributing to the females, basically how they respond to these drugs, and we are doing additional work with it. We map, this QTL is mapped to like 2 centimers or something. So the point is we can, so these synthetic populations I talked about the other day that we are experimentally evolving can also be used to map QTLs, and for me they're kind of similar things or different sides of the same coin, but other people don't see it that way. So, so anyway, this is my background. So the question is why did I get involved with the Peromyscus? Well, it's basically because of a web page I saw it. So here's the web page. If you go to the Peromyscus leucopus LL stock. So there's a stock center in South Carolina, which is where they have just maintained this stock of Peromyscus that was established from 38 animals, here circled, that were captured between 1982 and 1985. So I estimate this colony's been maintained for about 60 generations. And this colony was created just to have a source of P. leucopus. So it turns out like the military buys a lot of these mice from this resource center, and we don't know what exactly they're studying. And other people order them as well. And they're just basically a source of this mouse for physiology experiments. But to me, this is effectively our synthetic population resource, right? So it's, for me, in my mind, this is a 38-way cross that's been maintained for 60 generations. So it should look much like our yeast or our Drosophila, just with 38 founder chromosomes. So this is where we start to see, I think, here's an applied problem where many of the ideas from our evolve and resequence are starting to come into play, because this stock of Peromyscus leucopus doesn't look unlike the base population that Christian's lab's using for a lot of his experiments, right? It's just the number of animals that's varying a little, right? So a lot of the stuff, the way we think about things, should apply to this stock, I think, right? So that's why I really thought, wow, this would be really fun to look at this stock and do some work, right? So again, it's not perfect, right? So there's 60 generations of maintenance. Certainly the maintenance is at much smaller population size than we do in yeast and flies and C. elegans, but they at least made an attempt to avoid inbreeding. So similar to these other systems, there should be a lot of linkage to equilibrium in this system. 60, the number of years is such that although they've undoubtedly lost some of the initial diversity, there's still lots of alleles left. That's certainly true. Again, much like the work here, although I'm told that's changing a little, the founders are unknown, right, unlike our Drosophila and yeast populations. So the experimental strategy is pretty simple, actually. It's to take this population, this colony, and basically phenotype and genotype a large number of animals. So our initial experiment involves 300. I don't think that's going to be enough. We just measured a trait that was cheap to measure. It's a proof of principle initially. So the assay is we actually clip the end of the tail off, and that's commonly done in mice to just, you keep that little bit of tail and you can get DNA from it. So when we clip the tail off to get the tissue sample to make DNA, you basically touch the tail to a chem wipe, and you record the number of minutes until it doesn't leave a red spot anymore, which suggests, so it's basically a clotting time assay. It's widely used in mice too, I don't know why. And then our thinking was that, geez, given that this colony has much more LD and much less diversity than wild populations, perhaps we can genotype these mice like at pretty low pass, say 1x sequencing per animal, and impute the missing genotypes and do GWAS, right? So that's what I'm going to try to convince you that I think we can do. And I want to point out, though, that a problem with this system and an advantage of yeast is that the genome, remember, is 3.4 gigabases. So although we've sequenced about 400 individuals to 1x, that's about 18 paired and 100 high-seq lanes, or about $40,000 in sequencing, or $100 an animal. So this is clearly a lot. It's what I was joking to Christian about. This my Illumina habit is pretty expensive, right? So I could quit Illumina and take up heroin and save money, right? So it's sad. So, but there you go. But that's what I'm doing, okay? So clearly, as you get into these other, you got to be careful. If your system has a big genome, you have to find a way to raise money, right? It might involve hitting the liquor store once in a while. I guess there's a popular song in the U.S. about robbing a liquor store right now that most of you probably haven't heard. I'm living with 2 teenagers. So the program I've decided that we've been playing around with to do this is called Stitch. Not our program, was published by it's from Richard Mott's group in the UK. It's published in Nature Genetics. The main Robert Davies is the main developer behind it, though, and deserves the credit. Where they're trying to do genotype imputation from sequence without reference panels. So it turns out that although genotype imputation is this, if you just type it into Google, you'll get a zillion hits. It's used all the time in human genetics. Almost all the algorithms and methods are based on a world where you're in humans, where you have data from a SNP chip, and then you're going to impute and then you're going to have a reference panel, and you're going to use that to impute completely missing SNPs, right? Whereas in our world here, what we have is no reference and low-pass sequencing. So like each individual's genotype for a different set of SNPs, right? So the data is quite different. There's only a few tools for dealing with this data, surprisingly, right? So how's the method work? I found it to be extremely poorly explained in the paper, so I tried to explain it simpler. I don't know if I've succeeded or not, but for some region, imagine that there's actually F founder haplotypes that underlie that variation. So how the method works is it's going to say, well, let's basically for each individual, we'll imagine. So the data from an individual probably looks like what we're seeing here with this purple and with this blue and with this we're here where we have some SNPs are missing because it's 1x data. Most individuals are just going to get one SNP or one allele, and then we of course don't know phase. But what the method's doing is it's going to say, okay, I'm going to imagine most of the local variations due to these, say, 8 haplotypes. I'm going to lay down 2 haplotypes here. I'm going to find the best 2 haplotypes that explain the variation I see. And then I'm going to, it's almost like an expectation maximization thing where then I'm going to slightly modify the haplotypes to fit the individuals better. And I'm going to cycle through that like 40 times until I arrive at a set until I've invented a set of haplotypes that best explain the diploid data amongst the N individuals. And then I'm going to once I have these haplotypes, every SNP is known, so I can output haplotype doses and SNP calls for each individual, right? So what's clear about this method is more individuals, fewer true founder haplotypes, and bigger blocks or more LD makes it work better, right? So the sweet spot for this method is perhaps a synthetic population, right? And in fact, in the paper, they go and show that, in the paper, they don't even show using the method for less than 500 individuals here. So this is an outbred mice population where they do it on 500 mice. They do extremely low sequencing, like 0.1x, and they get extremely good imputation. Again, these outbred mice probably have much fewer founders and much more LD than our population. They also did it in a human population from Tibet. Again, they went with higher coverages, even more individuals, so the lowest number of individuals they use is 3,000. And they actually, by like 1x sequencing, they're getting fairly good imputation. And these human populations almost certainly have less LD and more haplotypes than our case, although you can see they're not even showing the graph here. So I suspect these come down pretty rapidly. So the trick is, again, feeding your Illumina habit, right? That you have to sequence more individuals, right? You got to get more Illumina data. Okay, you're hearing this a lot, right? Robert's the one who's got us started on the saying we need more, right? Yeah. No, for this, I think what makes the method work is that you can, is the individual data, because then in a sense you get the phasing and the haplotypes. Yeah, so sadly it's an individual-based method. But you're doing low coverage sequencing for each individual. Like, so in something like your population, if you had a method of cheap library production, you could probably go to like 0.1x per individual or even lower and get extremely good SNP imputation. So is this interesting? Is there people that are working in this group with individuals instead of pools? Probably, right? Yeah, more than one. So this is a neat thing to play with or think about, so hopefully. So again, but like we have to go, like, once we get into these applied problems, we have to sometimes move away from pooled populations, which are extremely powerful, but this is just the world we're living in, right? Yeah. These are not. These are, I use the Illumina Flex, and we have protocols to aggressively downsize the prep, and we just do it that way. I'll share the protocol. It's about $5 an individual for the prep. We found the Illumina Flex to be better than the old Illumina. It used the Nextera enzymes bound to a bead. I'll put my little Illumina thing on. And what happens is we can do the libraries in 96 reactions, and that we get fairly good representation across individuals. We found when we were doing plate-based Illumina preps with the old chemistry, we'd get a very high rate of dropout, or even worse, individuals that accounted for like a third of the reads in the plate, right? It seems like the Flex is a little better. I like the chemistry, yeah. They aren't paying me at all. I wish they were. I'll try to hit them up for money if I do a conference, though. They owe me some. My dealer owes me. Okay, so, no, but Shape-It's one of these programs that wants a reference panel, chip data, where you have say 500,000 really accurate genotypes, and then completely missing SNPs. Yeah. But I think with this, the stitch, it works directly on you just give it a list of your BAM files. So you just give it your, yeah.
I saw the numbers and we stayed away from it.
Yeah.
As you pointed out, the numbers are just 500.
Yeah, but what's cool, right, is that if we do another experiment, right, and sequence another 300 individuals, we can use the original 300, and like all the individuals we've ever looked at can be used to do the imputation, even if we're just looking at these 300. Like, so whenever we work with this population, every individual we've ever sequenced gets put in the database in a way. So, but yeah, it's scary, but yeah, but that's what but again, I think this is what we're once we go down the road of we're really interested in Peromyscus and Borrelia and Lyme disease, this is what we're stuck with. So I'm trying to show the trade-offs of taking our worldview and trying to move to an applied problem. There's a lot of neat, interesting, fun things, but you definitely pay a price, for sure. Like, I can't do as good a work. Yeah. Okay, so here's just some estimates of performance stitch. So what I've done, I don't know if this slide's that good or not, but I show it. This is just one chromosome 5, where I'm looking at so we don't have great measures of how we're doing with the imputation. So this is an experiment where we varied the number of founders from 2 to 20, and then we look at how well the error on our estimate of the genotype of the sequenced individual, right, where we have high coverage. So the sequenced individual, we know its genotype at heterozygous sites is known extremely accurately, and we can look at Stitch's error, right? Why is there an error? Because Stitch doesn't return like your genotype is big A, little a, or little a. It returns a dose, right? So it returns a number between 0 and 2, which is the number of non-reference alleles you have, right? So you get So a heterozygote might be 1, or it might be 0.9 or 1.1, right? So it's the best estimate of the dose. So what you can see here is obviously when we only imagine there's 2 chromosomes or 2 founder chromosomes in black, we do pretty poorly. But once the number of founder chromosomes gets like even above 0.6, it seems to be doing okay, although it certainly does better for some regions than others for reasons I don't understand. And that's basically the so why don't the number of founders matter? I think the problem is that although 20 might be more realistic, when the number of individuals we're looking at is only like 300 or 400, the 20 founder chromosomes is too few for the maximization to work, right? Because it's almost like there's too many degrees of freedom sort of thing. So that's a little practical problem with the software. So again, this is just the error. Again, another way of showing the errors, as the errors in the estimate of dosage is a function of the number of haplotypes. So again, once we get to something like, we're just using 8 haplotypes because it looks like the error rate's pretty consistent once the number of haplotypes gets to 4 even. But you can see the actual error per site can be quite high. Like, so there's a 95% of the samples fall within the heterozygote estimate of 1, plus or minus 0.8, right? Like, so again, a lot of at least in our current world, a lot of SNPs are estimated somewhat inaccurately, we think. So we think we have to grind through more individuals or do something else. But I also want to try to validate these SNP calls a little better. So I think we need a better way of validating the stitch output. I've been trying to do exome sequencing to get at that. Again, more samples would help. Even by the recommendations of the authors, n equals 400 is a lower bound. There's probably a reason they state that. But a feature of the approach is that new experiments can use old sequencing reads from other individuals to increase that efficiency. And again, we're hoping that as we get more and more individuals sequenced, we'll eventually be able to drop the coverage, right, is what the promise of the technique is, right? So eventually we should be able to drop the coverage. Right. We're only studying this one because we only have this one population. That's right. You can't apply it to another population. Yeah, or you got to start over. Right, exactly. But we do have SNPs now, and we can do genome scans. What's cool or sad or whatever is we have 17 million SNPs, right? So if you think of a human GWAS, a lot of the papers you've probably seen involve a 500K SNP chip, maybe a 1 million feature SNP chip with imputation. The human GWAS scans nowadays are using perhaps 2 million SNPs that they feel good about the imputation of. Well, in this system, we've just blown that out of the water. We're doing GWAS with 17 million SNPs. That also means we got to move our false positive threshold. So that's computationally difficult. So to do the scans, we've been using the software called GCTA, and particularly the MLMA routine. This software was written by Jiang Yang, who continues to support the software and work on it, but he initially developed it as part of the Vischer group, which is a big name in human genetics, right? So what we do, which is standard in human genetics, GWAS, we're going to correct for the first 3 principal components in the genotype matrix. You could argue for more or less. We're including some covariates. We measure on each mouse weight, age, and date of birth as covariates because they have in a linear model, these things all affect that bleeding time assay. So we just include them, although it actually doesn't seem to affect including them or not doesn't affect the outcome a whole lot. Okay. Yeah. No reason. I've done it with 8. It doesn't seem to affect it. I mean, at some point, as you add more and more PCs, you have more and you're burning degrees of freedom. I think the first PCs, I can't remember how much variation they explain in the genotype matrix, but of course that's dropping pretty rapidly as you add more PCs. There's definitely, Christian maybe started to allude to it a little bit, but stopped short. The colony, although the colony is this fixed thing, right? It's got a finite population size, so it's kind of like changing over time, right? The colony. And whenever you do an experiment, you're taking batches of mice, like you get a batch of 100 mice, and then 3 months later you get another batch of 100 mice, and then the mice come into your mouse house. You're paying $8 a day to keep a mouse. You try to do as many assays as you can, and then you sacrifice the mouse, right? So the way mouse genetics is done, there's the potential for this block effect, which is why we include date of birth. And then you want that genotype matrix. I think that's what the PCs are controlling for, that the fact that the colony is changing a little over time. The colony is also changing over time for a reason that should interest you. There's effectively, there's only so many breeding pairs. So at any given time in the colony, certain breeding pairs are producing a lot of the offspring that are being used. And then 6 months later, you might have another set of killer breeder pairs that are keeping the colony going. So there's definitely, the population is definitely structured, right? It's really cool, actually. So again, when you get into you get all these interesting things that start to pop out when you deal with the real world, I think. Yeah. Cool, good. Oh, sorry, the scan. So this is the GWAS on 17 million imputed SNPs, right? These are the 23 plus X chromosomes that, or 24 chromosomes that Peromyscus has, remarkably similar to us. I'm only showing you I'm not showing you the SNPs that aren't significant. I have a cutoff here, but it looks like there's some enrichment here on chromosome 1. It looks like there's something maybe going on here on chromosome 15. If we use the standard cutoffs here, so that's all rather exciting until we permute the phenotypes with respect to the genotypes in the panel and repeat the scan where we see some really significant sites that are, by definition, false positives. The permutations aren't perfect in this system because the individuals are related to one another. So I want to try to permute in a more sophisticated way, but this is, some ways conservative in the sense that we're seeing that with permuted data, we're also seeing these kind of things that you could convince yourself are hits, right? So I don't know where, so I'm not sure, how much I believe these, right, would be my point. So I can also go all Robert on the data, and I can basically take our 17 million SNPs, and I can basically make one of the SNPs a QTL that accounts for 5% of the variation in a fake character, and then I can rerun the scan. So here's the scan, the same scan where I've taken basically have a causative SNP that accounts for 5% of the variation that's right here. And so you can see, I think, a couple of things here that's important, that even if I have a single SNP that accounts for 5% of the causative variation, that it's not exceeding my threshold that would be rigorous to control for permutations, right? So there's a power problem, especially given that I'm doing 17 million tests. But you can see that there is this potential for enrichment, and you can also see that what we might think that because of the LD in the colony, there are several, like a block of SNPs that are probably that might be a megabase or bigger in size that are lighting up. So there's definitely information in there, and the method can definitely work. But again, I feel with 300 animals, we're at the edge in terms of our power. And then I also think there's a problem in that because our imputation of our genotypes has some error, it's almost like the sample size of the experiment's even smaller than that. No, that's right, it does not. Yeah, so this is even more optimistic, right? Yeah. So it's clear, again, to get a GWAS to work, even in a colony like this, you need to get the N up bigger than 300 mice, right? Which is, yeah. Yeah, this is just hide the QTL and additive 5% or BAM. One example, I could do 1,000. Yeah. But it sort of shows you that at least the assumption about the LD in the colony, meaning that you can get it with a tag SNP, is at least probably accurate. We just need more power, really. So again, the hide the SNP experiments are a useful tool, but they suggest more individuals are needed. Perhaps better genotype calls are needed. Perhaps bleeding time's highly polygenic, and we don't have a 5%er in there, which is why that GWAS fails. 17 million tests is going to be a problem that, is because, again, we're doing like almost 10 times as many tests as they do in a human GWAS, and so the significance threshold's an issue. But the tagging does seem to work. So the LD structure of the population is probably appropriate. Okay, so what about, again, like everyone's obsessed with SNPs and imputing SNPs, but we went over this the other day, right? That now we have to go back to our California principles. SNPs are just markers. Haplotypes change in frequency, right? So in this population, much like our synthetic populations, our belief is that there's really a limited number of haplotypes that are moving around. And the SNPs are really only marking those haplotypes. So maybe we have more power if we think about haplotypes instead of SNPs, right? So what Stitch actually returns, but you got to flip a command line switch to get it out, and no one ever flips the switch, is it actually returns these haplotype doses as well, right? So if we think of the data looks like this for individual 1, 2, 3, et cetera, if we assume a model with 8 haplotypes, their individual 1, Stitch might say this individual his dosage of haplotype 1 is 2 and 0 for everything else, which means it thinks this individual is a homozygote for haplotype 1. And this individual is a 1 for 2 and 1 for 8, means it thinks it's a heterozygote for 2, 8. And then this individual might be a 1 for 8, but a 0.5 for 1 and 2, meaning it's not so sure here. And then again, depending on how the algorithm works, these numbers might not, be close to 1 or 0, right? So that's what kind of the haplotypes are, right? That Stitch works with. And then of course, once it just uses these haplotypes to then bang out the SNPs, right? Okay. So what we can do is we can basically convert these haplotypes to basically dummy biallelic markers, right? So we can basically make a fake SNP, which is basically the dosage of haplotype 1 versus the dosage of all the other haplotypes. And then we can make that dummy SNP for haplotype 2, haplotype 3, all the way through haplotype 8, right? So we can make these dummy markers. Now, why do we do that? We do that because all the software that does these GWAS with 17 million samples that's extremely fast and efficient can't live with non-biallelism. It lives in a biallelic world. So all the stuff they do requires this. So what we're going to do is try to create these fake dummy SNPs that actually are haplotypes, but they're 1 versus everyone else haplotypes, and then run the scan with those, right? So that's an idea. Do you get that? Everyone get that? Yeah, it's kind of an idea. So again, we can run the scan. Of course, now we're doing 140 million tests, right? Which is kind of funny. And of course, we get what looks like look, there's something there. There's something there, there's something there. This is pretty exciting. The haplotype method's working, right? Except for, I didn't show it, but except for if I do a permutation of the phenotypes with respect to markers, I also get a few things exceeding that lot of 6 threshold. So again, I think there's a power problem, much like the other, that we have to deal with. But it's still an interesting approach. So I personally didn't like that approach, and so I have developed another one that looks at haplotypes. And what I'm going to do here is something a little different, where I'm going to, so again, I'm going to try to look at I'm going to take each individual, I'm going to classify them as basically a slow clotter in red, so individual 3 takes a long time to clot, and individuals 1 and 2 clot pretty quickly. So I didn't show you the bleeding time data, but it almost looks like there's these individuals that take a while to clot and another set of individuals that clot fast. So I decided to do this. This could be generalized to a quantitative trait, but right now I'm just doing it this way to kind of illustrate it, I think. And so what we could do is we could take all the individuals that are for some region and take all the individuals that are quick clotters and calculate their average haplotype frequency. So that's all I've done here. So this 1 here is the average of 2 and 0. This 0.5 is the average of 0 and 1. And this 0.5 is the average of 0 and 1. And I'm going to compare it to the average of all the red individuals, which in this case is just 0.5, 0.51. So for these haplotypes, I can just calculate the difference in haplotype frequency and make a score that's just the sum of the square differences, or what we call the Euclidean difference between haplotypes. So that Euclidean difference between haplotypes could be thought of as the total haplotype divergence between my fast clotters and my slow clotters, right? So that's a test that takes all the haplotype information into account, which we can't, the human geneticists don't do this, and they don't have software to do it, so I had to write my own. And so when we do this and run the scan, it's actually pretty interesting. We get an extremely large difference. And again, I'm not going to show you permutations, but we don't see anything like this in the permuted data set. The highest peaks are around 0.4. I don't think I show the permutations, but anyway. So this looks like something that maybe is sort of working. And again, if we go back, so again, I think I was fairly dismissive of power, because if we go back, so you might wonder, well, why does that work when you said there's no power? Well, if you kind of look at this figure, and instead of looking at individual SNPs, you kind of like smooth the data with some curve so that you're averaging over the actual LD in the sample. It does look like maybe there's some information there, right? So I think somehow this other test is exploiting that in a way that the SNPs aren't, because the SNPs are noisy, right? So this is a large difference relative to controls. So we can zoom in on this. So all I'm doing here is this is a big genome. Remember, we're talking a 3 gigabase genome. We have a peak that looks like, wow, that peak goes straight up in the air. It must be like a really small interval, right? So I'll zoom in on the peak. In fact, these Manhattan plots are extremely misleading. That peak is actually a fairly large interval. So actually, you could argue it's between, I don't know where, you guys can tell me as much as me, but here's like, say this is like 17 megabases to maybe 23 or something. So you could argue this is a 6 megabase window, which is, probably 6 centimorgans or something. So again, although the peak looks quite striking, the localization isn't maybe as good as we'd like it. Again, more individuals, I suspect, should help with localization. It always does. Nonetheless, we could take, because again, we've sequenced the genome and annotated, we can look at this whole region in the browser, right? So now I go into my browser for this region, and of course, this region contains a large number of genes. I can't remember how many, maybe 20 or 30 or something, maybe more. But right here at 19 megabases, so right kind of here, actually, or here. So again, I don't necessarily think that's a better candidate than that, but other people could argue. We haven't done simulations. There's a gene called CDC42. So this is a row GTPase, and if you type CDC42 or row GTPases into Google, you'll find out these genes are considered master regulators of platelet cytoskeleton and platelet function, right? So most of clotting has to do with these cells that somehow become activated because of a change in calcium, I think, or potassium, I can't remember. And then what happens when that happens? These cells explode and let platelets out. And these CDC42 is a master regulator of this process. So I'd say it's a very strong candidate gene that we can just zoom in on. one thing that's kind of interesting that's works follow-up is that there's this, so if we look at all the SNPs we've annotated just by sequencing wild individuals, there's actually a high effect SNP here, so that gets automatically annotated by a piece of software called SNPF, that you give it a VCF file and a gene annotation file. And this is actually, if I zoom in even further, but I won't, this is actually this actually predicts a SNP in a consensus splice junction. So you guys know in an intron starts with GT and ends with CA or whatever. Sorry, I probably got that wrong. I didn't teach genetics this quarter. But there's a consensus splice. It ruins one of those consensus splice dinucleotides that in vertebrates basically are at the start and end of every intron. So that would be a strong candidate for a SNP that, if true, results in this exon being left out of the message, which is interesting because it's an alternatively spliced exon to start with, right? So anyway, that's something we're going to do further work on that we think could be a hit. So that's again, or it could be a false positive, right? So I'm not going to claim that this is the answer. We haven't done the right experiment yet. I guess in my first talk and this talk, I tried to lean towards unpublished data because it's more fun to talk about and get feedback from, and for me, that's the spirit of stuff. So, but it also means that you got to put your BS detector on because some of this stuff might not be real, right? But anyway, that's what we're working on. And again, I think we can, but because we have a genome and SNPs and all this stuff, we can actually start to do some work in this system that wasn't possible 5 years ago. And I'm pretty excited about that prospect. So I tried to show you that I think there's a way to get to genes, especially if you're willing to consider candidate genes, and that's what we're doing. And again, it's future direction. So we have to do a lot more validation of stitch, but to do that we need markers from either sequence some individuals to higher coverage or RNA-seq or something, or exome. I tried an exome experiment where I used a human exome enrichment kit to pull down exomes from the mice, and that didn't work. I just got the data the day before I left. So I still hope exome sequencing can work, though. Again, all the stitch data, and I believe it, suggests that more animals should result in better genotypes, even if those more animals come from a different experiment. I'm writing a grant to do some Borrelia burgdorferi infection time course experiments. So the idea would be to infect the mice, a large number of naive mice with the Borrelia, and kind of do a pulse chase and then infect them, and then, because mice are expensive, and then basically do assays at various times after the infection to look at how, immune response and in tissue-specific ways and pathogen load in tissue-specific ways. And then from those animals, we would also collect DNA, and we could do a GWAS on multiple traits. Obviously, I'm working on improving these haplotype tests. We're also doing the genomics of antigen response, so we're actually just basically having the mice eat that vaccine I told you about, and then looking at how different animals have a stronger or weaker mount a stronger or weaker immune response to that antigen, and then looking at the genetics of that. Hopefully it's not all HLA. We're also trying to work with some collaborators to get CRISPR-Cas9 working in Peromyscus so we could test some of these candidate genes, but that's a little further out, I'm imagining. Acknowledgements in my lab, my tech, Polly. Jim Baldwin Brown was former grad student who helped with the assemblies. Yun Tao, current grad student who is actually really great. She figured out how to do all the high-C scaffolding, and she's kind of been doing a lot of that work. The UCI Genomics Core helped a lot, so we work with them very closely because we do a lot of Illumina and a lot of PacBio sequencing. The Peromyscus Genetic Stock Center, obviously this is only possible because they maintain this resource. It's pretty cool. The NIH has funded that stock center for 50, 60 years to maintain these Peromyscus stocks for the community, even though the community is small. It's an essential resource. My collaborator at UCI, Alan Barber, and his lab, Vanessa Cook, former postdoc, current student, Anna Milovic, who's, again, these are all people working with Peromyscus. Ali Mortazavi and some people in his lab to help with the genome annotation. Russ Corbett Ditag at UC Santa Cruz generate the high C libraries. The work's mostly founded by NIH, but it's pretty cool. We got this Bay Area Lyme Disease Foundation to give us $100,000 to do the genome assembly, which we burned basically on PacBio sequencing almost totally. But that was neat because they're actually more nimble than NIH, although they give us less money, because they were able to give us a pretty large amount of money on a pretty short timeline to do a very specific job. So sometimes that's a really neat way to use these private foundations, I think. And I can take questions. People stopped stopping me, so I do have quite a bit of time left. Yeah.